there, and welcome everyone to the first webinar hosted by Pepticom, a Jerusalem-based startup that develops AI tools to optimize peptide-based drug discovery. I'm Galia Koot, and I will be your moderator today. We're thrilled to see people from so many different disciplines and from all over the world joining us. And we thank you for all of the questions that were sent to us in advance. And our panelists today will try to address them. If you have any new questions, please add them to the chat. Israel is where the vaccination meets the startup nation. As of today, 55% of Israelis have received at least one dose of the vaccine against the COVID-19 virus. And at the same time, thousands of people here have been working on technologies to fight the virus and mitigate the effects. Today, we will be hosting a webinar on the subject, harnessing the power of AI to accelerate undiscovered treatments for COVID-19. It's my pleasure to introduce our first speaker, Professor Michal Linial. Professor Linial is a professor of molecular cell biology and bioinformatics at the Hebrew University of Jerusalem. She obtained her Ph.D. in molecular biology and has done her postdoctorate work at Stanford University. Professor Linial is the founder and chair of the computational biology department at Hebrew University. Her research uses large-scale technologies and data science from proteomics, genomics, and system biology, focusing on design principles in human health and disease. Professor Linial is working on the biology behind the coronavirus structures and is currently part of MAPAT, the Israeli government committee tasked with fighting the coronavirus. Today, Professor Linial will be talking to us on coronavirus cell entry, seeking the Achilles heel. Thank you.
Okay, thank you very much, Galia. I'm really happy to be here. And today, indeed, as promised, I'll tell you something about coronavirus cell entry and the Achilles heel of the virus. And I'll explain later on what I mean by that. So let's get started. So, okay. So just to put you in a state of mind of the virus, one can see here many viruses in yellow and the human cell in green, but besides the color, everything is as recorded by electron microscopy. And so for those people that say there is no viruses here, so there are many of those. So what is the state as of today, or as of March 2021? The entire globe is infected with over 115 million cases, 2.5 million deaths. And while we are speaking, there are 22 million that are actively with an active virus, which is quite amazing to think about those numbers. But let's go back to the beginning of the 21st century. And of course, at that time, we were introduced with SARS, and SARS of 2002 really infected over 8,000 people, which is less than what the number of deaths per day yesterday, let's say. But it infected 29 countries and started in China. It was a wake-up call because 10 years later, another virus from the same coronavirus big happy family came, which is MERS. And again, with few thousand of infected people and about 35% percentage of death. As you can see, the number of deaths was really high. But this is nothing to do or nothing similar to what we encounter today or those last year, where COVID-19 was introduced in the end of 2019 or January 2020, of course. This is a mistake, with a huge number of cases, and the entire globe is infected. So just to wrap up this intro, saying that we must, because of the scale, we must think about prevention, like vaccines, social behavior, whatever. We must think about medication, and I think we'll hear about it later on, and on some creative solution. I hope I'll introduce some of those. So we are looking for the Achilles heel because we are looking for the weak point in which we can, I would say, beat the virus, and for that, we have to understand how it gets in. Once it's in the cell, what's going on? What are the protein? What's the material that it brings into the cell? And a little bit, I'll tell you about the variants. So to start, like everybody likes to know, who are you? So in order to understand who are you, we better look at the many sequences that are there that are very similar. And the similarity just is indicated by this family tree that I think it's quite intuitive. Everything in red are those pandemic that I just mentioned. Everything in black are other animals that carry a very similar viruses. In this case, it's the bat, but as you can see, other animals carry other version of it. So if I'm just trying to do a compact view on the family, you can see that the three pandemic that I mentioned are here, but we have to remember that there are more of this. There are another 4 versions of the viruses that we call them kind of seasonal cold, you know, like a cute name, but they're really related to the family, but in a different branch of the family. And why I'm mentioning those, because it's very important to understand them, because they are giving us some clue on what will be the fate of those this SARS-CoV-2 that we are having now. So as I said, the protein, the virus carry more than one crown that everybody is familiar with. It has 29 proteins, which are look like that. So follow, just look at this. This is the 30,000 letters or nucleotide that compose the genome of the virus. And everything that we are talking about is this one, but it has 2/3 of it doing something completely different. And there are another accessory factors, 9 of them, that we know very little about. So let's zoom in into this S protein. But before doing that, I want to show you something very intuitive in my view. This is a list of bat genome. And whenever you see that the graph is up, it means that they are very similar. And you see that it's similar throughout the genome till it gets to a point in which it's not similar at all. This is maybe not surprising. This is the receptor binding domain of the S protein, which is the fastest evolving region in the virus. Okay, so once we say this, we want to understand how the virus gets in. So in order to understand that, look at this very famous, spike protein. And what I'm showing you here is the about half of the spike. And you can see that it has 2 colors, the light blue and the brown. The light blue are regions that are bind to sugar, very important, but usually we ignore them. The other part is the brown one, which is binds to ACE2. And for the more biochemists in the crowd, I would say that it's a very high affinity, which is about 10 to 20 times higher than the binding to the same, SARS, the original SARS from 2002. But let's look at the protein itself. It's a very well-designed protein, the spike. And when I say it's a well-designed, what I mean is that it's a braided multimer, as you can see, really linked to each other, very stable, full with sugar, as you can see here. This Christmas tree is really the spike with its sugar, and it goes through major conformational changes. Let me show you what I mean by that. The conformational change, if I take from the top view and I just flip it, you can see that at some point there is this jump. This is a big conformational change. And only because this change, the ACE2 can bind. And everything that I'm showing you coming from a cryo-EM, a very elegant technique to show molecular atomic view. So now let me just show you a few snapshots in which you see when it's relaxed, closed state, when one of those is open, as I showed you before. Now 2 of them are open, and only when they are open, they can bind to the ACE2. And this is once the infection is over, as you can see, it really changed its shape completely. So after understanding what I showed you as of now, we want to understand, can we find a really weak point? And this is the, from the Greek mythology, the Achilles, in which the only point that was sensitive was in his heel, and that was the only way to kill this guy. So this is maybe something related to this Achilles heel that I'm looking. And in order to do this, I want to introduce very briefly an experiment that we did in the beginning of the pandemic in March 2020, in which we wanted to understand whether the MERS that I mentioned before, or one of those cold virus, can tell us something about the binding. So whenever you see the result in this direction, it means high affinity, and here it's a low affinity. And immediately you can see that the COVID-19 and the SARS 2002 have very high affinity, while the other have very low affinity, and MERS have no affinity. So immediately we see that something is going on in the binding site, and this is what I just told you. So actually, when you look at the atomic level, you see something even more interesting, that despite that the two versions are similar to one another more than 75%, the patch that binds to ACE2 is completely different. COVID-19 binds here in two patches. SARS 2002 binds to a different patch. So why I'm mentioning it? Because it means that the procedure to bind to each of those SARS-CoV-2 or SARS 2002 or the cold virus is completely different methodology. Not only that, is that it's completely different number of amino acids that are interacting with each other. So let me summarize this binding story by showing you in a very simplistic way that ACE2 binds very strongly to COVID-19 virus. ACE2, the same ACE2, binds in a very different mode, a much more flexible mode to SARS 2002. And the cold virus only binds to few sites and in a very different direction. So the take-home message is that there is no shortcuts, namely the antibody for SARS that were developed will be useless. And that's exactly what was shown now after many months. So when I'm saying to the SARS, I mean the SARS of 2002. So once I introduce you this part, let me show you this weak point, which I think is a good Achilles heel point. What I mean by that, this is a point in which the only SARS, the only virus that has a site of cleavage is this one. And as you remember, in the beginning of the pandemic, people thought that might be engineered because how come that this is the only virus that has this cleavage site? But this cleavage site is very important because it leads to this conformational change that I showed you before. So just to summarize, activation of the spike happened in two places, on the surface, but also inside the cell. So if the virus was able to get in, there is this activation points that can occur there, and there is another one here. So everything that I told you up to now was on one single protein called the S. But what about the other 28? So let me show you what we know about them. The first 2/3 of them is dealing with replication. So, and with resource management. Think about this, that there is this amazing machine that is formed from 16 of the 29 proteins. And you can see it here. It's a sophisticated replication machinery. On some of those units, we don't understand what the machine is doing. But for example, this one is the replicase itself. This one is an error-correcting machinery, which is very important also for clinical point of view, but I won't elaborate on this. Now, because this very complex proteomics, people decided to look deeply into all the 29. And a couple of months ago, in a very famous paper in Nature, the following experiment was done. Let's take all each and every one of those proteins, infect a human cell, just by molecular biology, pretty straightforward, make sure that it's expressed in the cell, then just elute it with magnetic beads and ask by mass spectrometry what are the proteins that are bound. And what you see here is a snapshot of the result. You can see this is one in red, one of those proteins, and binds tens and hundreds of proteins. Actually, if I'm summarize this paper, we show that 330 protein were bound, and the idea was to do a repurposing of drug to those. But I mentioned this because I think it confused the community quite a bit. Why I'm saying it? As I said, because of the cryo-EM, we have bits and pieces of the structure, atomic resolution of many of those proteins, but we have it very partial. And what happened is that this experiment that I showed you before led to a lot of interactome, we call it, everything that interacts with the virus. But in my view, there are many faulty protein-protein interaction because a lot of aggregation, everything is done here in the outside of the cell. So we are worried about it. And for that, we designed with a group of partners in the Hebrew University, the following experiment. We take the same clone, the same plasmid, we infected cell, but now we are using a trick. And the trick is this, to start with this, we just cross-link proteins if and only if they are in a certain space from each other. So once we do this, we put it in the mass spectrometry and we got a protein with many, many constraints. So this is what we got. We got the proteins, and every time that we see a cross-link between two different parts, we say, okay, that means that these two different parts must be with a certain atomic distance with each other because it's a fixed space. And when we did this, you can see this green line. Green line are those cross-link. And immediately we were able to understand how the protein looks within the cell because we are doing it while within the cell. And when we do this, we identify direct partner, not indirect. And then we were able to see how this protein probably shut down the host translation system. And just to show you another slide from this same experiment or similar experiment, we took one protein which called NSP2. And the reason we took it is because we understood nothing about it. So that's a good rationale. But there was another rationale. This protein was resolved by Google just 2 months ago, or a month ago, by the AlphaFold DeepMind, and they came up with a structure that tell us that this is the structure. They did it with the AI, very influential work. When we took this structure and we tried to fit all those arcs, each arc is a cross-link. We couldn't do it. Everything was conflicting. And just to show you that every blue one is what happened within the cell. Every gray one is what happened outside of the cell. So just, without doing a lot of calculation, you can see that it's two different sets. But when we try to fit it, it doesn't fit. Only after reorganizing this protein, this is wrong. Only after reorganizing the protein, we could find the right solution within the cell because we were able to satisfy the distances of the crosslink. So this method is pretty powerful, and immediately we could find that new surprises for this NSP. We found that it binds zinc. Remember that in the beginning of the pandemic, people say take zinc. So I think it's a good idea, but zinc is an important parameter in the infection. We can also see that it has some kind of a electrostatic coloring pattern, which is different than MERS. And we are using integrative model, and the paper, it's a preprint, it's not yet published. So just to summarize this part, I want to show you that we have another set that we know nothing about. This is the accessory protein. Why I'm mentioning it? Because as I mentioned here, more question than answer. And why I'm saying it is because each and every version, like MERS and the Yuan version and the other one from Toronto and so on, you see that the changes are in this accessory. They change their space, they change their location, their name, and so on. This is a very important part that I think should be taken very seriously, but I won't elaborate much about it. The last part, the last 10 minutes or so, I'll tell you whether we have one virus or many different ones. So I'm going back in the beginning of the pandemic, and what I'm showing you here, this is patient one, the one from Yuan in China. And immediately you see that when you go from the center to the outside, you see that over time there are many clades that were evolved. And not only that, that each country have a different composition of the clades. For example, in Spain, you remember that Spain really got the pandemic in a very bad, but while Portugal has a different composition of the clades at a certain time. So fortunately, there is a database called GSED that by now have more than 5,600,000 genome that were fully sequenced, and we start getting a good view of what's going on. So many more variants, and the clade size is very sensitive to the sequencing coverage. And some countries just do not do sequencing, and we know nothing about them. Just to mention that in Brazil, for example, there are very little sequencing done, while in Britain, or UK, there are many done. And in Israel, I would say that in the last month, they started to do much more than that, than before, than in the beginning. So let me take you a few months ago. In the beginning, March 2020, really 1 year ago. At that time, you see by the color that this version from Yuan has changed. It's now become yellow and not green. That means that already in March, about a third of the viruses in the world were yellow. What I mean by that, it's a change in amino acids. And look at this, from January to June, 6 months. You start with a letter D, doesn't matter exactly. This is a letter of the alphabet of the amino acids. Start with D, and here at February, it starts to change. Now in June, within 3 months or 4 months, all the population of the viruses in the world have changed to a mutation here, which we call it D614G. And this change made the virus far more infectious. No one talked about it because at that time we didn't understand much about the viruses and the shape of the spike, but we already saw it, so nothing new by now. Now, of course, the game changer in the game was the production of vaccination, which against the original S is the spike. This is the antibody, they look like that. And it's very easy to understand why this protein, the spike, cannot bind ACE2. It just blocked. This is neutralizing antibody. Now, this is the many companies that are doing vaccine. We are familiar with Moderna, Pfizer, but we are familiar with many more. I just listed few of the most, those that are known to the public. But this is the virus that the vaccine was prepared for. Look what happened within a year, and look at those green, red one. Those red one are the British version, the New York version, the South African version, and so on. They are completely different from the original one. And this is just to illustrate this. This is the virus from the beginning to the end. In red is the spike. Everybody talks about those very important antibody, but as you can see, there are many other changes that are ignored. And just to mention one of those changes, which I like personally, happened in the spike, and it was already there in the beginning of the pandemic, but couldn't catch up, disappeared. And now it's picked up in the British version. And this is those, now you are familiar with looking at the spike from the top. Those are the important points that are changed, that were changed. And this is the same graph that I showed you before. Somehow in November or December, it changed completely, and the red one is the British version that by now is quite substantial in the world. So just to say that one clue for virus fitness, how good it is from the point of view of the virus, is to see that the same mutation reappears many independent time. This is a good sign, or bad sign, but this is a sign for fitness of the virus. So this is the South African version. From the top, as you can see, 3 very important mutations have occurred in the really very critical binding site for the ACE2. Actually, there are many more that everybody ignored, but I just show you on the top of the binding site. This is the South African. I should just finish by saying that it's a moving target. Why I'm mentioning it? Because we call those versions emerging strains. But there is a committee that calls some of them variant of concern. And the British one is variant of concern. Now I'm showing you a variant that didn't get this fancy name yet. It's called Q677H. And it appears 7 times independently. You see what I'm showing you? It's the yellow one. Independently, it appears. It were introduced around December 2020, and by now it's quite substantial. And let me bring you to the beginning of the talk when I mentioned this activation site. This is the site of activation. This is another important variant that I'm not talking about, and this is the new one, the 677. 677 is not that far from the activation site. And actually, if you look at it on the space, on the structural, you can see that it's here. It's here. And in my view, it pushes nicely the cleavage site, so the activation will be more effective. But as of now, this is a theory. I'm not saying that it's finalized. So to sum up the variant part, I would say that more sequencing, of course, will bring us more variant. Many of them will be with reduced fitness, not with improved fitness. I'm showing you 3 of the famous one. This is the Brazilian. This is from England, from Kent. This is South African. I didn't mention the New Yorker, which is a famous one also. And here, I'm just showing you the 3 variants of concern: the Brazil, the South Africa, and the UK. And you can see that at some site, all of them have the same changes. You see, some of them are unique. Just look by the color. Some of them too have the same change and so on. This is a very good clue to what's important for the virus itself. So just the last take-home message: the impact of the variant must be considered not through one by one, but through the combination of the changes, because there is no virus now that has only one change. All of them have many, as you can see here, really many. So just to say, everything that I talked about was on the spike when it's naked. But the virus, the spike doesn't like to be naked. Actually, with the dark blue, you see all those glycan that covers it very nicely. But this part, which is the binding site, it's exposed, of course. And why I'm mentioning this, because this variant binds here. This is the New York, by the way. And you can see that all the other places that not that many people are talking about are very close to critical glycan point, and might be that the shield of the sugar is something to consider. So the last slide saying that we cannot beat COVID-19 pandemic. we can beat, but not the virus. The virus will stay with us. And this is one question that was asked in the Q&A before, whether it's going to disappear. So I don't think it's going to disappear. I think it's there, but it will be reduced in its infectivity. We will develop some kind of immunity with the vaccine, without the vaccine, and it will be much, I would say, maybe in 5 years, or in 2 years, or in 1 year, like a cold virus that no one talks about anymore. But I just want to show you that if you look each and every change and you ask what will be the impact of it, we can see that this one that I talked about in the beginning, the outcome is much faster infection. This one is much tighter binding to ACE2. This one escape the antibody, the immunization somewhat. This one not only binds better, also more infectious, and maybe even has some escape from vaccination. And this one we just don't know yet. This is the last one that I talked about, the Q677. And by that, I think I'll stop, and I'll be happy to just to take. I mean, I won't take a question because I think I used the entire time. But there was one question that maybe I didn't answer that was sent before. That was concerning the whether. Why, how come that the MERS, for example, disappeared, and whether this is going to disappear, and this is the guess, you know. I think it won't disappear. It won't disappear because it spread to a level that it cannot disappear, because there will be island of infection throughout for quite a long time. We can see it in Israel, but definitely in the entire world. I think we should just be patient, and it will lose its fitness over time, as happened to many pandemic before. So I think it's a good point to stop here. Thank you. And I'll send it to Galia. Take over.
Thank you. Thank you very much, Michal, for a fascinating talk. This is very, very interesting. We also had a question, is this being recorded and will the link be made available? And it definitely will be. We are seeing already a lot of interest in that. As I mentioned earlier, Pepticom is a company that uses tools, develops tools to look for potential drug candidates for drug discovery. And about a year ago, we said, why not take our software and see what we can do with the new coronavirus? No idea what would come out. And the results have been fascinating, very interesting. And later, Amit and Immanuel will be talking about those results. But it really has been some year for all of us, all over the world, as we suddenly see that the world is really a small place. We're also trying to solve the same kind of problems, issues everywhere. And the virus is jumping from all of our countries. In the meantime, I would like to give a chance for Dr. Ariel Rokach to give us an overview of COVID at 1 year. What have we learned in this last year? What are some insights and thoughts? Dr. Rokach is a doctor of medicine and a master of health administration. He's the head of the respiratory unit at Shaare Zedek Medical Center in Jerusalem. Shaare Zedek has been on the forefront of fighting the coronavirus with over 3,000 patients, 200 who were in ICU, 9,000 visitors to their biological emergency center. And at the real beginning of the pandemic, Dr. Rokach was the head of the COVID-19 department at the hospital. Currently, he's involved with clinical trials concerning the coronavirus therapies. And it's my pleasure to invite our second speaker, Dr. Ariel Rokach.
Thank you. Good evening, good afternoon to others, good morning to others. We'll share the presentation. One moment. Okay. Okay, so COVID-19 is 1 year old. What we have learned? We have learned a lot. The main thing that we have learned is that we still do not know many things. This is the first slide, and it might be also the last slide also. So, Professor Linial gave a wonderful speak about the virus. The next 30 minutes will be about the patients. I'll speak a little bit, very shortly, about the history, then about the strategy to control the pandemic, and mainly I'll focus on treatments. And after me, of course, the researchers about new medications and new horizons will speak about them. So this is the tiny virus, 100 nanometers, but it changed the world. It changed the world this year. So the history starts with two cousins that Professor Linial has already mentioned. The SARS that first reported in Asia on November 20, 2002. And actually it disappeared in 2004 and nobody knows why. We don't know why it disappeared and COVID-19 was spread all over the world. But we know that it was a real threat to the world. We know that the death rate of SARS was almost 10%. Much more than COVID-19. And another frightening thing is that around 21% of the patients were health workers. The second cousin of COVID-19 is the MERS that emerged in 2012 in Saudi Arabia and is called Middle East Respiratory virus. And actually, most of the viruses, most of the patients that were infected with MERS had a severe disease. MERS had a rate mortality of almost 35%. 35%. Imagine to yourself that we had such a numbers in the COVID-19. It means dozens of millions, dozens and dozens of millions of deaths all over the world. We were lucky that the death rate in COVID-19 is much, much, much lower. The MERS also disappeared. Again, we do not know why this disappeared and the COVID is still here. Just an anecdotal article that was published 14 years ago by researchers from Hong Kong. Actually, they saw the future. Look what they have written 14 years ago. They wrote that the presence of large reservoir of SARS-CoV-like virus in horseshoe bats together with the culture of eating exotic mammals in Southeast China is a time bomb. Therefore, the need for preparedness should not be ignored. But we ignored, and the world was not prepared well. Okay, so we'll start with COVID-19. It appeared in Wuhan at late 2019. This is the reason for the name COVID-19. And rapidly, the outbreak infected actually all over the world. And on March 2020, only 1 year ago, the WHO declared that COVID-19 became a pandemic. A few weeks later, more than 1 million people were infected all over the world. And a few days later, more than 100,000 people died from the virus. Really a deadly virus that spread dramatically all over the world. Meanwhile, we have more than 100 million people that were infected, more than 2,500,000 deaths, death rate of around 2% all over the world. In Israel, a small country, almost close to 10% of the population was infected. Almost 6,000 deaths. The death rate, a little bit lower, 0.74. But you know, this number is very hard to judge. If we, for example, had measured all the population, the mortality rate would be very low. If you had tested only the symptomatic cases, the numbers would be high. If we look at the numbers in a population that the age is older, the numbers would be high. In a young population, it would be low. But around between 1 to 2% of the patients might die. And in such huge numbers, it's a very high number. So, how to control the pandemic? First, we'll speak about strategy. I chose 5 elements. I'll speak about each of one of them very shortly. Masks, social distancing, epidemiological investigations, PCR tests, and lockdown. A few words on masks. Exactly 1 year ago, the Israeli Ministry of Health said masks are not effective against COVID-19. A few days later, we knew it was wrong, and now we know that masks are very important to control the pandemic. But we still have problems. You know, it took weeks to months to have enough N95 masks for healthcare workers. It took weeks to months to have enough masks for the general population. And many still do not use the mask properly, and many do not use them at all, even though we all know that it is very important to wear to control the pandemic. A few words about social distancing. It seems logic, it looks simple, but 1 year after the beginning of the pandemic, it is very problematic. It might cause loneliness, depression, lack of education, unemployment, and the harm from the social distancing, some can say that it might be higher than the damage of COVID-19 itself. I still believe that social distancing is important, but we must be aware that there are very problematic things also in this thing. Epidemiological investigation is a very important tool to control the pandemic, but still we're not there. We made a great progress, but still we don't do it good enough. Still, it's a problem all over the world, and if we improve, we probably will control the pandemic much better. PCR tests. It took months to increase the capacity, but we have a lot of questions. Should we screen all the health workers every month, every week? Maybe we should screen all the population. What about false positive? What about positive tests for remnants of viral RNA that confused many physicians? So we know that PCR test is a very important tool, but still we do not have all the answers and we still have many questions. Lockdown. Is this the solution? Probably it worked. This is the main thing that worked. It worked in China, it worked in New Zealand, but is it really the good solution? We're not sure. At the beginning of the pandemic, people say, look at Sweden. They controlled perfectly well the pandemic without a lockdown. But look now, they themselves admit that they made a mistake. And it's a country around 10 million citizens, very close to the numbers in Israel, and the death rate is more than double. So did they succeed? I'm not sure. In New Zealand, they locked their country. They did a lockdown for one single patient. Is it good? They succeeded, but I'm sure that they have many other problems. So it is still a question how to control the pandemic. We have certain measures, but we do not know for certain what is right to do and what is wrong. Now we'll focus on treatments. First of all, we'll speak about the healthcare workers that they're treating the patients. Then I'll speak a little bit about supportive treatment, respiratory support, because this was the main problem with the patients. Then we'll speak about antiviral drugs, drugs against the virus, and drugs against the inflammatory response, because later on, when the patient is deteriorated, we need another treatment. The virus sometimes is not there, and we need to treat the inflammatory response. I'll speak a little bit about blood clots and about complications that need usually antibiotics for secondary infection. Okay, so we'll speak about health workers. At the beginning, 1 year ago, we were afraid. We thought that it might be very dangerous for the health workers to work. And actually, many were infected and many died. But if we take the good measures, we usually be safe. So at the beginning, when we were concerned and we had lack of protective gear, we said that only minimal medical stuff should be inside with the patients and more stuff should be in the control room. But we know that it was a misconception because COVID-19 patients need help. They need supportive treatment. They need supportive environment. And because we changed completely the conception, now we know that COVID-19 departments should have more nurses, more respiratory technicians, more physiotherapists, more ancillary staff, more volunteers, more physicians than in a regular medical and ICU department. We also know that the patient needs their family. At the beginning, we didn't let in the family, but we know that it is important for the patients and for the family to see each other. So actually, we changed completely the concept, and now we are all inside the department, working, helping the patients. We know that the main problem of the patients, the main problem are the lungs, and the oxygen is maybe the most important treatment for the patients. Low flow oxygen, high flow oxygen, invasive ventilation, non-invasive ventilation, and also the ECMO, all those are supportive treatment that are crucially important for the patients. And why they need it? You here see a chest X-ray with all those infiltrates on the right and on the left. This patient is suffering from bilateral pneumonia, from ARDS, and he cannot breathe, so he needs help, respiratory supportive help. Now we can see a CT with such infiltrates. And here we see another problem, blood clots in the main arteries of the pulmonary arteries, and they also are a very important thing that we have to help the patients with this. Okay, so on March 2020, it was declared that severe COVID-19 patients should be ventilated as soon as possible. And it was declared that non-invasive ventilation might be harmful for the patients and might be very dangerous for the health workers. And now, a year later, March 2020 now, most of us believe that the opposite is right, and that non-invasive ventilation should be started in most cases, and it is safe for most of the patients, and it is not dangerous for health workers. And invasive ventilation might be harmful. So we also now change concepts. And at the beginning of the pandemic, because we thought that severe COVID-19 patients should be ventilated, most of the countries bought thousands of ventilators. The Ministry of Health in Israel told us in 1 month we'll have more than 5,000 Israelis that might need to be ventilated. So we spent hundreds, millions of dollars to buy ventilators that are useful, that we don't use now. But now we buy high-flow nasal equipment, high-flow nasal cannula that helps the patients. And now we know that this equipment has already saved lives of thousands. And we now know that high-flow nasal cannula is not dangerous for health workers. At the beginning, we didn't use it because we thought that it might spread aerosol of the virus, but now we know it is not true. Okay, so we spoke about a supportive treatment, about oxygen, invasive, non-invasive ventilation. Now, treatments against the virus. Treatments against the virus itself and treatments against the inflammatory response. Two different treatments, but both of them are very important, and also treatments for blood clots and treatments for complications. We had many drugs that were given to patients. I will not speak about all of them. I chose only few and a few words about each one of them. Hydroxychloroquine on March 2020, study from France showed a beneficial effect. The FDA approved it, and actually in the first 3 months of the pandemic, I think that most of COVID-19 departments all over the world treated their patients with hydroxychloroquine. But only 3 months later, we knew that it has no benefit, and maybe it might be harmful. So it might cause QT elongation, it might cause arrhythmias, and it might kill the patients and not help them. So the FDA that approved it on March disapproved it 3 months later, and I think that nobody treats now COVID-19 patients with hydroxychloroquine. What about anti-HIV drugs? They were effective against SARS and MERS, and it was a big hope on March 2020. But only 2 months later, in an article on the New England Journal of Medicine, it was stated that there isn't any benefit of those drugs. And actually, they're given now only in clinical trials and not routinely. What about anti-IL treatment? It's not against the virus, it's against the cytokine storm, against the inflammatory response. Preliminary results show that it might be beneficial. But an article in the BMJ 2 months ago said that it might increase mortality. So nowadays, we use anti-IL-6 only in clinical trials and not regular use. What about plasma from recovered patients? It seems perfect. We take plasma with antibodies from patients that recovered, that could beat the virus. So logically, it would help the patients. So the FDA approved it, and we gave it to patients, and we were sure that we're helping them. But 6 randomized trials in hospitalized patients couldn't demonstrate a clear clinical benefit. And actually nowadays, less and less it's given to patients because we know that we do not have a good proof that it is beneficial. What about antibodies? It's logic that it will help. But here we have a dilemma. Usually COVID-19 patients deteriorate after 7 to 10 days. At that point, the main problem is the cytokine storm, and antiviral treatments at that point probably would not help. So should we give the antiviral treatment at the beginning? But at the beginning, 95% of the patients are mild patients and they don't need this treatment. So antiviral drugs should be given only for high-risk patients at the beginning of the disease. And this is what is done nowadays, and we hope we will succeed. What about remdesivir, a novel nucleotide analog that is interfering with the virus, treatment against the virus? It was a very big hope at the beginning, medication that helped for other diseases, for Ebola, for example. We were sure that it will be the great treatment, but we are still not sure. A very big meta-analysis published on the BMJ with over 7,000 patients could not prove that this medication is effective. But later on, a final report in the New England Journal of Medicine gave us some hope. And actually, we saw that those that received remdesivir had a median recovery time of 10 days as compared with 15 days among those who received placebo. A very important thing in the days of pandemic when a lot of patients are hospitalized and we have lack of place for patients, shortening of the hospitalization is very important, but we could not prove that it saved life. Now, the dexamethasone, steroids, treatment not against the virus, but against the inflammatory response. Actually, this is, I think, the only treatment that was proved in several trials to reduce mortality. Actually, it showed reduction of mortality of about 20%. The placebo patient in one study, the death rate was 40%, and the patients that were treated, the death rate was only 32%. So a real reduction of around 20%, and other studies also showed it. So actually nowadays, in most COVID-19 departments, we treat patients with dexamethasone, and we believe that we reduce death rates by this treatment. We saw that many patients have blood clots. So preventive therapy for hospitalized patients is given with anticoagulation. Of course, we have to be aware from thromboembolism and give full treatments in certain cases. But probably the best treatment is prevention, as we know. And we saw many, many treatments that failed. We were sure that many of them will be a great success, and they failed. But we know that the vaccine is working. This is an article published 1 week ago by our colleagues from Clalit HMO. they did a real-life study on more than 1 million people. Half of them got the Pfizer vaccine, half of them didn't get it. And they saw a very good protective effect of more than 90% and protection against mortality. It proved again what the first studies showed, this vaccination is working. And if we vaccine most of the population, probably we should solve the problem. But we're still not sure, because we have also questioned about the vaccination. Should we vaccinate children? Should we get the vaccination every year, every 6 months, every 2 years? We still don't know if it's effective on certain mutations. Maybe we'll have to develop new vaccinations, and maybe we live like this and get vaccination every year. We still don't know. We have a lot of questions with the new mutations. At the beginning of the pandemic, we knew that children and pregnant women are minor patients. They were never been severe cases, but nowadays we know that more and more pregnant women and more children develop severe disease. And it also might be changed with the new mutations. We have questions how to treat post-COVID-19 patients. We don't have time, so we will not deal on this. I'm involved in one study, so I'll say a few words about it. The aim of this study, the aim of this trial, is to evaluate the effect of a food supplement called NLC. This food supplement has an antiviral effect by inhibiting the 3C protease, and it also has anti-inflammatory effect on IL-6. The primary endpoint of this study is to measure the length of hospitalization. We don't have results yet. We don't know if we succeed or fail. Only the future will tell. So, what we have learned in the last year. We have learned a lot about COVID-19 in the last year, but the main thing that we have learned is that we still do not know many things, as I said in the first slide. We have learned not to draw clear conclusions too quickly. Many said many things at the beginning. Oh, it's a common flu, it's nothing. Not more than 10 people in Israel will die, don't be panicked. But we know that they were wrong. But at the same time, it is sometimes important to act quickly, even when there is not enough information. We have to admit when we are wrong. I'm sure that I was wrong many times. And we have learned from the virus, we have learned modesty. Okay, I think that my time is over now. There were many questions. Maybe I'll only answer one question that was on the treatment given in Ichilov, a CD24 inhaled medication. Actually, the idea is great. Inhaled medication that stops cytokine storm. It looks like a perfect treatment, and it was really a success, but we must be careful. It was only a phase 1 study. We have to do another study with a placebo-controlled trial, and then we'll see if we can repeat the marvelous results that we saw. Okay, thank you again. And now to the future. We have a pandemic, but we still have hope. We have many patients, we have many dead people, but we have hope to succeed. And the other lecturers will tell us about this hope and about the future.
Thank you, Dr. Rokach, for a very interesting and comprehensive talk. I like that you ended on a note of potentially hope, and I like to look at it more as optimism. Optimism when I think that, first of all, things have to get better, but also optimism when I see all of the technological advances developed this year and people working together all over the world to try to find solutions to help combat this virus or to fight the results and the effects. And with that in mind, I'd like to welcome Dr. Immanuel Lerner, the CEO and our host, the CEO of Pepticom. As I mentioned before, Pepticom is a company based in Jerusalem that's building tools that use AI techniques in peptide-based drug discovery. Immanuel received his Ph.D. in biochemistry from Hebrew University, as well as many years of experience in research. He has published papers in many of the leading journals. And today, as the CEO of Pepticom, he's leading the charge here in the company in research, growth, and funding. Immanuel will be talking to us on harnessing the power of AI technology for peptide-based drug discovery. Immanuel?
Dyna, thank you very much for the introduction. Share my screen. Okay, as the CEO and founder, co-founder of Pepticom, a company that is basically a leader in AI-based peptide drug discovery, I will talk today about harnessing AI technology for peptide-based drug discovery. The talk itself is going to be less technical. We heard excellent reviews from Dr. Ariel Rokach and Professor Michaeli about the biology of the virus and about treating patients. And this is extremely interesting and teaching us really what the world is being experiencing. One thing for sure is that we need more tools in our toolbox as we don't know what the future holds here. And AI can be one of the driving forces in discovering and helping us develop new potential drugs for this outbreak, for new mutations that may be facing us in the future, maybe this year, maybe next year, who knows. Hopefully, we will not be experiencing another great outbreak, but we have to be ready, as the world cannot really will withstand another huge wave of a new virus, as far as we know. So in Pepticom, we believe that we need to integrate not only AI, but computational chemistry and biochemistry together when we work on the discovery using AI of new potential peptides. What you can see here in the movie is actually a project that we have done, an ongoing project in Pepticom, where we discover in silico using the AI technology potential peptides. Then we synthesize them and early develop them. And I will show you that this approach is extremely helpful in many ways when it's compared to other current technologies that are not AI driven. Pepticom is a post-round A company, and we work in two main modes. We either work in-house with our own projects, and we collaborate with pharmaceutical companies and also with some agricultural companies. So you may be shocked to know that for every dollar put in R&D in pharma, 85 cents are lost due to failures. This is unacceptable in any other industry, and it's caused by many different factors. Most of them are just not enough knowledge of biology, of medicine, of chemistry, and a very tight regulation. When we are facing a pandemic like COVID-19, we really need to be able to discover and develop drugs much more efficiently than what we do today. And AI can hold at least part of the answer for this. And we are sure that in the future, this is what is going to happen. And when we are thinking about AI, we can take computer vision as a good example. We all know, or we all saw the "I'm Not a Cat" video that was just published a few days ago, a few weeks ago. And there's the very known example of the chihuahuas and muffins. Basically, what we see in this application of AI for computer vision is that this is a solved problem. Since 2015, computers are basically better at recognizing pictures than humans. And this was done by deep learning technologies. And the answer is, okay, so if this is solved, what is not solved? And what is really not solved, and where we really need AI, or it's the next frontier as far as AI is in drug discovery and development. One of the biggest applications, I think it's even more important than distinguishing between chihuahuas and muffins. And what you can see here is basically the way drugs are discovered and developed today. Pepticom is situated in the phase of target to hit up to end of preclinical testing. And what is important to understand here is that when we are thinking about applications of AI in drug discovery and development, first we need to understand that there are many different phases and many different problems you need to address using the AI technology. So you need to fit AI exactly in the right, answering the right problems in every different part of this very long and complex development process. The second thing that needs to be clear is that we don't have a complete knowledge of the problem. So we still lack knowledge in chemistry and biology and medicine. So the applications of AI are not trivial. What you can see here is that basically when you start a discovery effort of a potential drug, you start with the genome sequence, which is now completely solved. You have then the huge problem or challenge of identifying a potential target. For this, it was usually done in academia or big companies trying to find new potential targets. Today, we have AI applications for that. Cytoreason, an Israeli company, is doing exactly that, helping us using AI to find potential targets. Then structure, Michal mentioned DeepMind, Google's DeepMind ability to basically take a gene sequence that we can access easily from every computer available and fold it. So now folding or having a structure which is linked, which is basically a target, is something that we can do easily. We also have huge amounts of new information from cryo-EM and other technologies, giving us really an unprecedented access to structure of proteins, which is really key. Now, this is exactly where Pepticom steps in. We can take this information and discover active peptides that are drug-like or drug candidates extremely fast and more efficiently than other ways of discovering potential drugs. And I will show you exactly how in a second. And then, of course, first of all, there are other companies doing drug discovery, most of them not in using AI, not in the field of peptides. And worth mentioning that in clinical phases, there's also use of AI, but in completely other aspects, more about trying to fit the best patients and the most efficient way of driving these very difficult and costly clinical phases. Something that is very important to understand is once we have good applications of AI into this very long and risky process, it will probably, first of all, make this process much more efficient. It will probably change the regulation and will change the way the pharmaceutical industry actually works and the way we discover drugs and our health. So before talking about what Pepticom does, it's important to understand what peptides are. So peptides are basically very short proteins. And proteins basically run all the processes that we inside our cells. And the interesting thing about peptides is that they can serve as drugs, and Amit is going to talk about it in the next session more deeply. They can be basically used for many different aspects, but one very interesting is trying to inhibit two proteins from interacting one with the other one. It's called protein-protein interaction inhibition. And this is very important, and I mentioned it because the ACE2 and spike proteins from the human receptor and the SARS-CoV-2 virus actually need to undergo this protein-protein interaction, and peptides could be a good candidate for trying to inhibit this and basically serve as potential drugs. But how do you do it? How do you find peptides? So it's extremely difficult to find active peptides, and there are many technologies trying to do that, and I'll give an example a bit later. We are taking an approach which is inspired by nature. We call it swarm artificial intelligence, where you can see here a flock of birds. They sense the environment, they interact with each other, they reach conclusion, and they share the conclusions. And basically, in analogy, what we do in our technology is three things. First of all, using these software robots, we map the protein problem. We learn about the area in the protein where we want to target our peptides. Then we use reinforcement learning in order to design potential peptide drugs ab initio from scratch. We don't use any library to do that. And then there is a discovery process where peptides are discovered in silico and they compete with each other until we get the best peptides. These are synthesized and tested in our lab. We have a whole branch in the company doing biochemistry and biology, testing and validating and helping us improve on the peptides, because once we have an active peptide, we also need to optimize it, as in any drug discovery effort. So this is basically what we are doing here. And one of our benefits when we're using AI is that it can have a lot of features when you're approaching a drug discovery project. So we cover a huge chemical space, 10 to the power of 30. It's 1 with 30 zeros afterwards. It's impossibly long. We know where we bind, and we have a very large diversity as far as the chemistry. And this is important because when you're trying to find a new potential drug, you always, one of the first things you do is finding an active molecule that is actually active on your target, okay, of interest. But that's not enough. You need the target, the peptide, or any other molecule to be stable. And what our technology enables is that because we have a very large chemical diversity and ways to make the peptide cyclic, it's not a linear peptide, we get attributes of stability. And we did some work. You can see here in the graph in the bottom right side. We did it together with an English company called Interact. You can see a peptide that was discovered using our technology. So this is an active peptide. You see that it's completely protected from proteolytic activity in blue versus a linear natural peptide that could be discovered using other technologies, which is degraded within half an hour completely. So the big benefit or the big promise of AI technologies is that we would be able to add more and more features into the discovery phase, reducing the failure rates afterwards, because we will be able to control at least some of the aspects that are important for drug discovery. This could be also toxicity and other aspects of drug discovery and development. Here you can see an example which is for us very, very exciting, and it's linked to the pharmaceutical industry. So there is, here you can see data from a target that was, I cannot disclose it right now, but a very known target, protein interaction that the pharma industry was looking for inhibition of this target for over a decade. Many efforts were done with large libraries of peptides, over 10 to the power of 11 molecules. So teams of scientists spent many years trying to solve this, and there were a few successes. What you can see in the top right is one such success from a company that used this 10 to the power of 11 different molecules, and you can see an active peptide, which is very nice. We used our technology with half an employee for less than a year and got the same potency of peptide. The interesting thing, first of all, is that we did this with orders of magnitude less resources. But the important thing here is that our peptide is cyclic and non-natural, which makes it much more suitable as a potential drug, and the development of this molecule would be much easier. The top molecule was never developed as a drug because it didn't have the right properties. So regarding the SARS-CoV-2 and our efforts in this arena, what you can see here in the movie is the human ACE2 protein, the human receptor in light gray on the bottom side, and on the upper side in darker gray, the virus spike protein. And if you can see, you can look in the interface, you can see a green and a blue molecule. These are peptides that were actually discovered using our technology. When this pandemic started, we had the technology already running and alive and kicking, and we just couldn't help ourselves trying to play with our technology and see if we can find something that could be of a potential inhibition of this virus entry into cells. And we actually ran the software, we synthesized the peptide, we got some help from a pharmaceutical company for that, and tested the molecules, and we got very interesting results. This is now a subsidiary company of Pepticom that was just funded very nicely for an early project like that. And now we have a company that is completely focused on trying to find potential inhibitors of viral entry into cells. And we'll discuss in the next presentation why we think this is very important. So this company already has validated the peptides. We will show you some of the prototypes in the next peptides in the next lecture. This company has already validated screening methodologies and a SARS-CoV pseudovirus screening methods using biological safety level 2 facility that we have, and we are working in the Hebrew University using that. So what I showed you here is the ability of AI to really revolutionize the way peptide drugs are, and drugs in general, are being discovered. I didn't enter into too much details as the audience is very broad and with different technical knowledge. So I try to give the simple short version of the things. Next after me, we'll have Dr. Amit Michaeli, our CTO, talking, and he will review in more details what we do. And it's going to be a more technical talk. And so thank you very much. I will just, I have here one question. Just see. Okay, so there's one question I got. Have you found it easier or harder to access data from various sources to fuel your development efforts? So it's important to understand that we use reinforcement learning, and we do not need large data sets of potential peptides or peptide drugs. Anyway, there are not many of these. So our approach doesn't really need large data sets for that. We do need data, and this is Amit maybe can address exactly what kind of data we are using, but that's not a hurdle for us. For us, the important thing is getting the right structure and knowing where to target the specific structure in order to target or to initiate a discovery effort. So thank you very much, and feel free to contact me if there is any interest from your side. Galia?
Thank you. Thank you very much, Immanuel. While Immanuel is the front of the company, the front of Pepticom, Amit Michaeli is the man behind the magic. And as the CTO and co-founder of the company, he's developed the technology used by Pepticom to discover new drugs. Dr. Michaeli has a Ph.D. in computational chemistry with a specialization in peptides and protein design. He also has degrees in human genetics and an MBA. And right now, Amit will talk to us on implementation of Pepticom's technology in the COVID-19 pandemic. Amit?
Thank you, Galia. One sec. Okay. Can you see my, I can't make it for some reason. All right. Sorry. So I'm just going to cover our discovery effort and the ideas behind our methodology implemented in this pandemic. And as Dr. Rokach and Professor Linial showed us and talked about in more depth before, this is not the first coronavirus we're encountering in the 21st century. Prior to this, we had the lower spread but higher mortality SARS and later on MERS, with about 10 years between them. And then about 7 years later, we got COVID-19. And we know that mutations are like lottery tickets. The more viruses we have, the more random mutations to undergo, the more likely we are to get new strains. And Professor Linial showed us the new strains that are evolving very rapidly in SARS-CoV-2. And we hence see this as a problem that's here to stay, unfortunately. We do not think that this is going to disappear. SARS was a warning sign. SARS-CoV-2 is already a serious global pandemic, and we should be ready for SARS-CoV-3 and other variants to come along in future years. And I think that this is the enemy of humanity in the 21st century in a manner that's kind of similar to the Tower of Babylon story from the Bible. Humanity got together in a globalized economy. We're all working together towards improving ourselves, but local epidemics quickly become global pandemics. And unless we have a good strategy of dealing with this, globalization is going to have to end. And in fact, right now, our airports are all closed. It has been halted until we solve this problem. And our vision for how we should deal with COVID-19 is a multi-layered defense. This is not a new term. It exists in medicine. It exists in the military. It exists in the martial arts. And the idea is that if the virus has to go through several layers to create a lethal pandemic, the chances of that happening are much, much lower, and we should have normal lives. And the three layers are, first and foremost, vaccination, which is thanks to the Israeli government ongoing very rapidly in Israel right now. And this should be protecting the mass of the population from the virus, where the second layer should be antiviral drugs. It should be multiple drugs that cover multiple targets, as was exemplified by the successful HIV cocktail. This will help us avert escape mutations. So the chances for a virus strain to escape one drug are low, but the chances of escaping 5 or 6 drugs administered simultaneously are much, much lower. It's the probability to the power of number of drugs. So we're talking about a much, much lower chance for escape mutations. It needs to be able to be administered in a large scale and be of relevant cost. It can be extremely costly because this will not work for global high population pandemics. And the third layer is what was talked about by Dr. Rokach, is cytokine storm inhibitors. The reason that we think this is the third layer, and this was also shown by Dr. Rokach, we don't really know what the effects of messing with our immune system at different stages of the disease will bring. So we know that patients undergoing a cytokine storm respond very well in the phase 1 clinical trial to cytokine storm inhibitors, but we don't know whether this will help patients that are in earlier stages or may actually aggravate the disease to go worse. And of course, if we wait till the patient becomes critical, as also Dr. Rokach shown us, we will have many complications arising from the patient's critical status, complications such as clotting, pulmonary fibrosis, etc., that we don't want our population to have. So this is the third layer of defense. And as Immanuel stated, Pepticom specializes in peptide drugs. And peptide drugs are an interesting concept because they're a middle ground solution between large proteins and monoclonal antibodies and the classical small molecule solution. Whereas small molecules are often only limited to dealing with concave targets, such as enzyme pockets. Proteins can deal with almost any kind of convexity, and peptides are a middle solution. They can deal with more than small molecules, but less than monoclonal antibodies. And also when we talk about oral availability and toxicity, which small molecules are naturally more toxic than proteins, peptides are a middle solution with a mid-level toxicity and mid-level oral bioavailability, but that's improving. And how is that improving? The next generation of peptide chemistry is weaving away from the natural amino acids. So our classical peptide drugs that currently are being administered at the hospitals around the world are mainly composed of the 20 natural amino acids. Of course, our bodies know how to metabolize those because they exist in our proteins. Mostly they metabolize through the N and C terms of the peptide. So the classical peptide drugs are non-cyclics. And of course, because they're so metabolically unstable, they're limited to intravenous injection. And that's a major problem. Today's peptides are moving towards cyclic-sized peptides, as you can see in the example inside us. So this formed the cycle much more metabolically stable because the N and C terms are not exposed. And as you can see in this actual peptide that we're working on, out of the 5 different residues, 4 are completely non-natural, stuff that our enzymes in our body have never seen before and hence will be less likely to metabolize. So we hope that with these changes, we keep the advantages of peptide drugs in engaging a more versatile set of targets than the classical small molecules or the existing peptide drugs, and yet give them something more of a stability like the small molecule counterparts. So where do we fit in the corona pandemic? Of course, because we're not going to engage small pockets or enzymes that exist because small molecules can do this probably better. Our main advantage is dealing with the spike ACE2 interaction. Again, Professor Linial has already shown this to us in quite depth. So we can see the spike protein here in red. It basically connects to the ACE2 on the human cells. And this pretty much starts the process of fusion that allows the virus to enter the cell and multiply itself and create a pathology in the patient. And our idea was that we should basically find peptides that bind spike and prevent it from attaching itself to the ACE protein. This is not a concept that we just invented. This is a concept that's actually been deployed in the HIV cocktail. We can see Mariavoc on the left that inhibits CCR5 and enfuviratide on the right, or T20, which also binds in the same manner. And the idea is that the one on the left is actually a small molecule that you can see that has a very deep pocket. And the one on the right is in fact the peptide that inhibits HIV fusion. And this idea is very good for peptides to be deployed in, because I'll show it in a second. Peptides have one distinct advantage over proteins. So, and this is before all the stuff that was in Professor Linial's talk was known. We started this in March, but as a general strategy, if you look at the monoclonal antibodies versus a targeted peptide as a fusion inhibitor, we're talking about a less potent solution. So a peptide will work well at 50, 100 nanomolars. That's exciting. Again, there are cases where peptides work at lower dosages, but it's a very different size molecule we're dealing with. So it's going to be more difficult to reach a potency quite like monoclonal antibodies with a typical potency in the single-digit nanomolars. But peptides have an advantage, and that advantage is shown here in the circle. Assume that we assumed in March last year already that the RBD domain that you can see here in green is going to be mutating. And in fact, Professor Linial showed us that it is indeed the most hypermutating area in the coronavirus. And then when you talk about a mutation, a resistance mutation, something that will escape either vaccination or monoclonal antibodies, then as you can see, and this is an actual antibody that's been solved in the crystals, PDB7DD2, if it can form any mutation within the black circle to the right, and basically push out this monoclonal antibody. So while it's very potent in binding, one steric interference, so for example, take a valine, put a tryptophan in there, much bigger amino acid, may push the whole thing away. Potency is not going to help for sterics. And the interesting thing is that in this black circle, it can actually push the antibody while retaining the binding to the ACE2 that you can see here in yellow. So an area that's not exclusive to the ACE2 binding area is a great area for the RBD mutation to push out this monoclonal antibody. And we feel that still that this is where resistance mutations will most likely take place. There are mutations in the ACE2 binding interface, but for a mutation to be effective in the ACE2 binding interface, where you can see our peptide on the left, it needs to do two things. It needs to push our peptide out, but keep the ACE2 in. So it has to go through two probabilities, two different lottery tickets. One, to push our peptide out, and two, to do this in a manner which retains in the same area, in the same physical space, ACE2. So we think this is a great strategy for developing a drug that will have a higher resistance for mutations, and that's what we went for. Before we also went for this, while we were going for this, great academia publications came out, and they took a pretty simple approach. What they did, and the Baker team and Prote team did two interesting but similar strategies. They took, in the case of the Baker team, they stabilized mini proteins derived from the human ACE2. They stabilized them so they're very strong in their 3D structure. And they showed the ability to bind spike and become very potent inhibitors. Whereas Prote et al. Took peptides from different corona members in the same manner and showed that they're able to inhibit by competing for the human ACE2. And we can see that in pseudoviruses, they reach inhibition levels of around 70%, 75. So this is very interesting and very exciting, but these peptides are not very good drug candidates. They're mostly composed of the natural amino acids. They're very large. They'll be difficult to produce. They're great academia proof of concept, but we want to go towards drugs. And what we did was focus our design libraries on small peptides under or equal to 15 amino acids. We wanted them to be protected from N-term and C-term metabolism. So they're all cyclic. They're all head-to-tail cyclics. And their composition is heavily leaning towards the non-natural subunits. Over 70% non-naturals. And in fact, the example Immanuel showed you for metabolic stability was taken from one of the initial prototype peptides that also binds the spike protein. So these things are small and metabolically stable. And because they're also small, they're going to be reproducible within reasonable cost to serve as drugs. And what you can see here is the actual case of the AI learning how to bind the spike protein. You can see that it converges pretty nicely into lower energy levels. Lower energy levels mean better binding. So this is our combined energy function. And the peptides then reach a minimum. And then we want to see that this is in fact the optimal solution. So what we do in reinforcement learning, we take out all the smart bots that learn really quickly and perform another set of learning. This is to differentiate fast learning from good learning. If this was only fast, then we would get another wave here, which is lower, and you'd see several waves as it goes down. But in this case, in fact, we see a smaller wave. So the bots that learn fast also learn smart. And just to give you more of an intuitive, this is the molecular weight of the different solutions. And you can see that they also, while this process converges, lean towards the bigger molecular weights, because this is, again, a pretty flat protein-protein interface. So this would be a human intelligence check of AI. So this is something any medicinal chemist would tell you. And in fact, the bots figured it out themselves. Another interesting to see is this is what the solution set looks like. They're backbone cartoons. And you can see that they exist in different clusters. You can see one pointed by the arrow. And these clusters are showing us that the bots actually converge onto several solutions, but each solution is a family of solutions that are pretty close to one another. I'm showing you stuff from our prototype peptide. We've already advanced more in these clusters. And the first thing we did, because we just didn't have everything sitting because this was a pandemic, was test whether they bind the spike RBD as we anticipated. This is an example of one of our early prototype peptides and tested using microscale thermophoresis or MST. This gives us the binding affinity to the spike protein, and we see a hit level binding affinity of 3 micromolars to spike. We then had alpha LISA prepared and tested these peptides using 2 different assays, alpha LISA and ELISA. Alpha LISA tests whether the peptides can separate 2 fluorescence probes, one sitting on the spike and the other on the ACE. If they separate the probes, then we can say that the peptide disturbs the spike-ACE interface, and we can see 2 peptides marked in the A block. And then we have ELISA, which shows complete separation of the protein complex. We can see that in B. We then had the great opportunity to work with the team of Dr. Michal Galtanami from Bar-Ilan University. And she tested these peptides on a pseudovirus. And but basically these pseudoviruses express spike. They have a signal in them that shows us that it was able to infect a cell, and it's tested on human cells that express ACE2. And we show that one family in distinct, which you can see here in blue and red, had nice blockage of this. Another family was shown a bit weaker here in yellow, but we have new families and extensions of these families coming forward. And you can see that this actually shows a potency which is pretty close to the KD level. So this means that actually being in a cellular solution made these peptides more potent. But one thing that's interesting is we only reach about 50–60% inhibition. So this creates the big question mark, and we'll talk about it in a few minutes. First, we saw that the limited inhibition was also exemplified in pseudoviruses by Prote and et al., which is a very nice published paper. So this is not an artifact that was done because of the performance of our experiment by the Tanami group, but probably something which is more likely systematic to this screening assay, because the same peptides at Prote et al. Also showed 100% inhibition of the virus. So we're now taking some of these prototypes to actual virus testing to see whether we see the same phenomena. Where the pseudovirus only shows limited inhibition and the virus, the peptide actually inhibits the virus completely. We're also working on bicyclic derivatives, which will make the peptides more rigid and hence more potent and even more stable. And we're doing post-AI structure activity relations the good old classical way, using our eyes to make these peptides go better. And we also have an anti-ACE2 binding program, which will be an inhibitor on the human side. And because the human is not mutating as quick, could be more mutation resistance on the way. So thank you very much, everyone. And I'm open for any question.
Right. Thank you. Thank you very much, Amit. That was great. And I'd also like to thank everybody who's attended our webinar today. And we hope you found the content very helpful. Thank you to our speakers, Professor Michal Linial, Dr. Ariel Rokach, Dr. Immanuel Lerner, and Dr. Amit Michaeli. If you have any questions or comments, please be in touch, and we hope to host you again. I also wanted to thank all the institutions and companies that have collaborated with us on the work that we're doing right now. Please, everybody, keep safe and healthy, and thank you very much.